Let me begin by describing the overall procedure of this lab. We're going to be making a genetic change in the bacteria E. coli using CRISPR to make a point mutation in its DNA. Normally, E. coli is vulnerable to the antibiotic streptomycin, but the mutation we'll make will prevent streptomycin from recognizing the E. coli. To demonstrate this, we'll create 3 petri dishes that contain agar. Agar is like a jello. It's a substance that bacteria can grow in. The first petri, this one here, will only have agar, and we'll only use it to grow a lot of bacteria for our experiment. So we start it off and let it grow up. We have to take some time for that. The second 2 petri dishes also contain agar, but they also contain streptomycin. Streptomycin, which normally kills the bacteria. So here's our strategy. First, we grow a large amount of bacteria in the agar plate. Then we harvest the bacteria by splitting it into 2 separate parts. One part, we just take a little bit of it and we put it right straight into the agar with the streptomycin. That's a control group. The second bunch of agar, we put into a tube and then we subject it to the CRISPR materials so the bacteria are made immune to streptomycin. Then we put these bacteria into this second, third petri dish right here. Now we wait to see what happens. The experiment worked. What should happen is the bacteria in this colony here should grow. The bacteria here should not grow because this bacteria has been made immune to streptomycin and this one has not. But supposing that didn't work out, it didn't work that way. Supposing, for example, the first petri didn't even grow at all. That probably means that the bacteria, the E. coli, were dead on arrival, or you didn't wait long enough to let them grow. Supposing neither petri 2 or 3 grew, neither one of them grew. Well, this one didn't grow because the streptomycin killed it. And if this one didn't grow, it means that the transformation we made with CRISPR didn't take. So it died of natural death just like these guys because the CRISPR didn't work. The third possibility is that both of them grew. Okay? That means that the streptomycin wasn't active. It means that the E. coli can grow here because the strep wasn't active and it grew here for the same reason. And I think those kind of will lead you to where you have to look to see what the problem was with the experiment. So now let's get on with the actual details. So let's begin by making the agar. You're going to receive a tube that looks like this from theOdin. And this is the tube that contains the agar, LB agar, but it does not contain the streptomycin, okay? Don't make a mistake of using the wrong tube. Okay, so you start off with this tube here, and you go to this glass jar, and what happens is, take the cap off, you just pour this into the glass jar. I'll take the top off here, and you probably should use a funnel to do it. And there it is, we're going to pour it in. And that's it. It's in. So now the agar is in there. Next thing we want to do is we want to add water, 150 milliliters of water. Basically, they also send you this measuring tube here. And we're going to put in 3 of these because they're 50 milliliters apiece. And then we put the agar into the microwave, and we want to cook it for about 2 or 3 minutes at 30-second intervals. Don't boil it, and don't put the cap on tight. Make sure the cap is loose, okay? And after the agar cools down a little bit so that you can grab it, then pour the plates. Just pour enough on each plate to cover the bottom and get as many out as you can. I'm going to probably get about possibly like 7 plates. Let's see what happens here. So we'll pour this one first. Okay, the second one. The third one. Fourth one. Fifth one. Sixth one. The seventh one. And there we are. So we've got 7 plates. They're all filled. Okay. Now what I want to do is I want to put the tops on them and just let them cool. That's it. So we just let them sit there, cool down, and the tops are going to get steamed up because the agar is putting off water. And after they cool down enough, I'm going to flip them over and put them in the refrigerator for later use. Okay. Once the agar is set up and gelled and is solid, then we can add the E. coli. From Odin, you should have got a little vial that looked like this that contains agar, and you can see a little stab, a little vertical white stab through it, which is where they added the E. coli at Odin. So what we're going to do is we're going to stick an inoculation loop into that and drag some of that E. coli out, and then we're going to plaster it onto the agar right on the surface. So we don't want to break the surface. So here's what we do. First of all, we open up this guy here. Okay. Then we take the loop and we try to follow the same track that the original track right there. Try to follow that track and bring it out and see if we can get some. And I'm going to wipe it gently on the surface of the agar here. And I'll get another little dab of it. And I'll wipe it on the other one. Very gently so you don't break the surface. And what we're going to do is we're going to grow this coli so that we'll have a lot more than the small amount we have in this little vial here. Now what I want to do is, now that I've put it in, I'm going to cover them and turn them upside down. And I'm going to put them in the refrigerator in one of these little bags here. Now I'm going to put them in the refrigerator at, not the freezer, but just the refrigerator, to let them sit there for a while. It's possible you can leave these things out all night for 24 hours all night to watch it grow. Or I'm just going to put it in there a little bit and then I may take it out later. But that's the whole point. Now what we're going to do is wait until the E. coli grows. And now while the E. coli is growing on the agar, it's time for us to make the streptomycin petri dishes. And so we take this tube of LB strep agar and we mix it up with 150 milliliters of water. Same procedure as with the regular. Now we add water. "50 milliliters at a time. And so you put it in the microwave, and as I say, we cook it up for a few minutes, 2 minutes at 30-second intervals. And now after letting the agarose with the streptomycin cool off a little bit, I'm going to pour them into the petri dishes for our next part of our experiment. I'd like to show you what I did was I marked the bottom of the petri dishes with what was in it. This is my symbol for agarose plus streptomycin. And the reason why I didn't put it on the covers is because when you take all the covers off, you might put the wrong covers on the wrong petri dish. So by marking the bottom of the petri dish outside, the outside bottom, you are sure that you have the right petri. So here we go. I'm going to pour this agarose with the streptomycin into these petri dishes here. Here we go. Again, as they predicted, we get 7 petri dishes out. And I'm just topping these off here. So we get 7 petris out, which contain agarose and streptomycin. Now I'm going to put the tops on and let them just sit here for a while until they cool off a little bit. As they steam, the steam is going to rise onto the top of the petris, as you can see. And now I'll just let these set, and we're going to these are the second set of petri dishes, which are the ones that will have the control. Only one of them is going to have a control, and the other one's going to have the CRISPR transformation in it, and we'll see what happens to the bacteria. The next step in the process is to see if we have grown any E. coli. So there's the coli. You can sort of see it, the stripes there. We've had it growing for 48 hours in the agar. So now we have more E. coli we can work with. So the next step of this thing is I want to transfer some of this E. coli to a control petri dish which contains agar plus streptomycin. This is the control, okay? So the first thing I do is, this is a petri which contains agar plus streptomycin. I open it, and then I open this agar with the coli in it. You can see it here. So what I'm going to do is I'm going to scrape some of this off, some of this coli off here, to get a little bit of it in the tube. And they're going to transfer it over here to the agar and streptomycin. There it is. The transformation is complete. So, the next thing we want to do is put the top back on this guy, and on the bottom I'm going to say that I've added the coli to it. Plus E. coli. There it is. Now I'm going to let this grow, but it's not supposed to grow because that's the control group. And now we're at the stage where we have to do a little more delicate biochemistry. You've got to be pretty careful at this stage, and you have to have everything kind of set up. To begin with, what I have is the original agar and coli. I still have that colony there that I can draw some coli from. And then I have the agar and streptomycin in this one right here, which is where I want to put the after I do the transformation with CRISPR, I want to put it into that petri right there. Okay, for now I'm going to leave it aside. So this one right here with the agar and coli, what I have to do is I have to take this agar, this coli rather, and put it into this little tube here, which is empty. And then what I want to do is I want to add these guys one at a time into this tube right here and mix them up. Okay, and I have to be really careful because I'm using a pipette here, and the problem is if I use the pipette more than once, or even once in either one of these things one at a time, I'm going to contaminate the pipette, and it's going to contaminate the materials. So what I've got to do is I've got to get a tip from here, draw this out, put it in here, then I have to discard that tip, get a new tip, draw this out, put it in here, discard that tip, and do the same thing repeatedly 4 times. So the basic idea is that I've got to be extremely careful about being sure I don't contaminate anything on the way. This is why you have to be sort of methodical here. Okay? To begin with, this is where the original coli are living. I've scraped some off already, because I've used it in this over here, which is a control. Over here, I have this control out of sight from you, but that's a control version where I've simply said, okay, put the E. coli, without doing any kind of CRISPR transformation, put it into the streptomycin and agar and see what happens. What should happen there, it's this one right here, what should happen there is the streptomycin should kill the E. coli and nothing should grow. But this one over here is going to be a different situation, because we're going to use these guys to transform the coli that comes out of here. And we're going to transform it so that it becomes impervious to streptomycin. Then when we put it into this petri right here, which contains streptomycin, the E. coli should grow. In other words, the raw coli doesn't grow here, but the transformed coli will grow here. So without anything further, let's start off. I'll follow the recipe, the same recipe that you guys are using. And this is the original coli right here, growing in the agar, and there it is on the bottom right there. Okay, so to start off with, what it asks for is 100 microliters of this PEG transformation. So we have to get 100 microliters of this stuff right here, PEG transformation. So I pull this out. First of all, the first thing I better do is I'm going to set my to 100 here. There we are at 100. I'm setting my pipette to 100. I'm plunging it into one of these pipettes right here, tips right here. Lifting it up. I've got the tip now. Now what I want to do is I want to open up this peg. The peg is the first one. I'm going to open it up. And I'm also going to open up this micro centrifuge tube here. Everything's going to be put into here. So what I do is I press down on the top of this guy right here, drop it into the peg, and let go very slowly. And if all goes well, which it did, I've drawn up some of the peg. Now I'm going to take that 100 microliters of peg and I'm going to squeeze it into that tube right there. That's done. It's all in there now. Now I want to discard this. You press the other button, wherever you're going to discard it. Press the button and it flipped right out, of course, naturally. But so that's taken care of. Now I've completed that part. The next section of the process is I have to get some of the ecoli out of here, and I want to put that into here. So to do that, I have to get one of these inoculation tubes here. And now what I do is I say, okay, let's get some of these guys and gather them up so I can put them in here. So I start dragging along where they're growing, trying to get a bunch of it onto my. It's doing pretty good. I'm getting a bunch of it. That's really good. Looks good. So I get some here, as much as I can gather. See, as much as I can get. Okay. Now I want to put this into here. It's very small, so you probably can't see what's going on very well. There it is right there. It's kind of hard to make it let go. Get her to let go there. There we go. It's letting go now. It's not easy to do, let me tell you. I hope I got a lot of it out of there. Yeah, I got pretty much all of it. Okay, so that takes care of that. Now what I want to do is put this back over here. Now I want to begin getting 10 microliters of each one of these, one at a time. First, the Cas9 tracer RNA. That's the first thing. So I have to go down to 10 microliters. That's the first part here. I believe that's 10. Yep. So this is set at 10 microliters. So I grab another pipette tube, like so. Open up the Cas9 tracer RNA. I want 10 microliters of that. So I press down on the top, drop it into the container, let go. So there I have it. I have 10 microliters of that. I squeeze it into the open tube. Takes care of that. Close this. We've got some more in here I can use later. Discard the tip, like so. Time it landed in there. Then I want to get the second one, which is the gRNA. I want 10 microliters of that. Okay, grab the tip here. Squeeze the top, drop her in, Go. It's got it. And I inject it into here. There we go. Discard the tip. The third and the final one. Open that up, which is the template DNA. Grab a tip. Press down the top. Drop it in. Let go. Sucks it up. Put it into the tube. There it goes. Discard the tip. And close this. Now, the reaction is now taking place in here. So I close this thing. And what I do is I'm going to leave it 30 minutes in the fridge. It's mixed up pretty good. I'll let it 30 minutes in the fridge. That's the first step. In the last 5 or 6 minutes, before you take the E. coli out of the refrigerator, by the way, it's not in the freezer, it's just in the refrigerator. What we want to do now is, they also gave you some little tubes of agar, and I'm going to add some water to the agar. I'm going to shake it up to get a bit of agar in here, which we're going to pipette some of this out and put it into that mixture. And then we're going to let it sit for a while to incubate. And finally, we're going to wipe it onto that Petri dish with the streptomycin. Okay? So I'm going to mix some water in here. And I'm just going to fill it up, and I'm going to shake it up a little bit just to get it so that we have a little bit of agar, some food for the bacteria when they come out of the refrigerator. Okay? Okay, so what we want to do now is this little bath here is approximately 108 degrees, and I'm going to take the stuff in the refrigerator. I'm going to put it into this bath for 30 seconds. That's to give it a heat shock. That'll help the DNA and the CRISPR and the Cas9. It'll help everything to get into the cell, sort of open the cells up a little bit here with the heat shock. Okay, now what I'm going to do is pipette a couple of 100 microliters of the agar we just mixed up a few minutes ago into, that would be nice, that would be handy. Okay, here we go. $100. $200. I'm going to give it a tiny bit more, a little bit more, be sure we're okay here. I'll give it about 250 maybe, something like that. And there it is. And now we've got the food in there, we've got the CRISPR in there, we've got the E. coli in there. Now what we're going to do is we're going to incubate it from 4 to 6 hours at room temperature. I'm just going to leave it right here on this corner, and I'm not going to do anything to it, and we'll see what happens. And now that the E. coli that's been treated with CRISPR has been incubating for 4 hours, or actually 4 hours and 15 minutes, now I'm going to put it onto the agar with the streptomycin to see if the CRISPR will prevent it from being harmed by the streptomycin. So I'm going to mark this plate here with CRISPR. So we'll know that that's the plate that's being treated. So there it is. I want to open up this guy here. And I want to take 200 microliters. First I'll grab a tip, and I want to get up to 100 here. Okay, there'll be 100 microliters of these. Here. 1, 2, and I'm going to stir it a little bit to make sure that it's spread out pretty good. We'll see if we can get it to grow. So if all goes well, what we should have here is a plate full of E. coli. See what happens. And there it is. Now the only thing that remains is for us to let it sit and let it grow. We'll see what happens. And now the moment of truth with the experiment, which was obtained from theOdin.com. There's the control, and there's the CRISPR. Let's find out what happened. The control, no E. coli grew, as you can see. It's still just as blank as it was before. The agar is just sitting there blank. But with the CRISPR version, you can see that the E. coli grew very well. So the conclusion is the experiment worked, and the conclusion is that the CRISPR actually worked on the E. coli to change its genetics so that it was able to survive in streptomycin. That's it. That's the conclusion.